Welcome to This Explainer. Today, we're diving into something truly mind-bending. We're going to explore how scientists are basically taking the structural rules of documentary scriptwriting and using them to literally rewrite the genetic code of life. I mean, we're talking about an incredible journey here, going all the way from some pretty basic agricultural tweaks right up to literal life-saving medical miracles.
Okay, so to kick things off, consider this fascinating insight from our sources. While mathematicians study what is and physicists study what probably is, bioengineers are doing something completely different. They're actively building entirely new biological stories that never existed in nature. They study what never should have been. And they're doing this by learning to write the very code that dictates how living things operate. It's wild.
And this brings us to a really brilliant comparison. Just as a documentary filmmaker blueprints a sequence before they ever shoot a single frame of video, bioengineers write and edit the code of life to program living cells. Think about it. A filmmaker creates an outline, right? They structure their narrative into acts before heading out into the field. Well, similarly, a bioengineer designs plasmids, promoters, and specific DNA sequences to structure a biological narrative before putting it into a living cell. It's like they're the directors of biology.
Let's move right into Section 1, Printing and Programming DNA, from atomic gardens to synthetic plasmids.
Now, to really appreciate where we're at, we got to track the evolution of this technology. We're moving from the low-stakes world of farming right up to bleeding-edge medicine. Historically, humans edited crops using incredibly slow or, frankly, totally chaotic methods. For thousands of years, it was just selective breeding, slowly turning basic field mustard into entirely different foods like cabbage, cauliflower, Brussels sprouts, the whole shebang. But later, in the atomic age, scientists literally placed highly radioactive pillars in the center of circular gardens just to randomly mutate the seeds around them. No way, right? But that chaotic, radioactive process is actually how we got the vibrant red grapefruit you see in the grocery store today.
Fast forward to today, and thankfully, we don't rely on random radioactive chance anymore. Scientists deliberately program custom DNA plasmids using 3 exact sequential steps, much like structuring a script's 3 acts. First up, you pick an origin of replication, which essentially tells the cell's machinery to grab on and start copying the DNA. Second, you add a selection marker, like a gene for antibiotic resistance, so they can easily weed out the failures and keep the successfully modified cells. And finally, you insert the functional code, a promoter to start the read, the specific sequence you want, and a terminator to tell the cell, okay, cut, stop reading.
So the crucial point here is what happens next. This custom script gets installed into bacteria, like E. coli. And E. coli is a fantastic biological factory because, get this, it has a rapid doubling time of just 20 minutes. Plus, its membrane makes it incredibly easy to modify. Once that custom plasmid is inside, the E. coli reads the mRNA instructions exactly like an actor reading a script, and it starts producing brand new custom proteins. Scientists actually routinely program these bacteria to produce stunning, fluorescent proteins that glow super brightly under UV light. Absolutely incredible.
All right, let's jump to Section 2, editing out the errors. Transitioning from DIY synthetic biology to life-saving human medicine.
Because let's be honest, making glowing bacteria in a lab is cool and all, but the stakes become monumental when we need to edit out a fatal error in a human being's existing genetic script. To do this safely, doctors are now utilizing something called base editing. This is a revolutionary molecular tool in the CRISPR family. But instead of traditional gene editing, which acts kind of like molecular scissors that break the DNA strand, base editing is much more like a precision word processor. It can safely correct a single genetic typo, literally changing one single DNA base to another without ever severing that delicate gene end.
And that leads us perfectly into Section 3, a life-saving breakthrough. The amazing story of KJ.
We can really see the profound emotional impact of this script editing power when we look at the real-world case of KJ. He was a baby who desperately needed a personalized medical intervention right after he was born. KJ was diagnosed with CPS1 deficiency. Now, this is an ultra-rare genetic metabolic disorder. It affects only 1 in 1.3 million newborns. Talk about a needle in a haystack. Because of one specific typo in his genetic code, KJ lacked the vital CPS1 enzyme. That meant his body couldn't convert toxic ammonia into urea. And without an immediate fix, this rapid ammonia buildup causes severe neurological damage, seizures, coma, and can be completely fatal. Conventional treatments just weren't cutting it.
Time was absolutely running out, so a massive collaborative team of academics and clinicians pulled together and achieved what I can only describe as a medical miracle. A 6-month speed run to rewrite KJ's genetic script.
In month 1, they analyzed his genome to pinpoint the exact specific mutation causing the issue. Through months 2 to 5, they designed a highly customized CRISPR base editor tailored exclusively for him. And then in month 6, they implemented a 3-dose lipid nanoparticle protocol. This was designed to specifically target his liver cells, since that's exactly where the CPS1 enzyme is normally produced.
And you guys, the results were nothing short of astonishing. Just weeks after those lipid nanoparticles delivered the base editor directly to his liver, KJ showed immediate clinical improvements. He was suddenly able to process protein. He required a significantly lower dose of his ammonia-reducing meds, but most importantly, he began hitting vital physical and cognitive developmental milestones that this disease had previously threatened to steal from him. Which brings us to Section 4, the future of medicine, expanding the genetic editing toolkit for humanity. This incredible proof of concept is rapidly pushing the boundaries of genetic medicine. We're already seeing this technology scale way beyond just ultra-rare infant diseases. For instance, a Louisiana man recently became the first in his region to be functionally cured of sickle cell disease using these exact same advanced genetic techniques. This perfectly proves that KJ's case is not just an isolated miracle. It's actually the beginning of a massive shift toward treating a wide range of debilitating genetic disorders right at their source code.
But of course, we have to pause and think about the big picture here. Scripting our own biology requires some profound ethical oversight. We have to carefully balance this crazy rapid innovation with strict safety protocols and total transparency in clinical trials. As these personalized therapies become more common, regulators, scientists, and industry leaders are having to collaborate globally. They're basically building international regulatory frameworks from scratch to make sure these incredibly powerful tools are used safely and responsibly.
So that leaves us with a pretty massive, profound thought to end this explainer on. We've gone from selectively breeding mustard plants to instantly rewriting a baby's genetic code to save their life. We have crossed a massive threshold here. As these biological word processors become more advanced, more accessible, and even more precise, what happens to humanity when we finally possess the power to fully rewrite our own biological scripts? It's a future that's arriving way faster than we ever imagined, and it's definitely one we all need to be ready for.